We're going to be talking about Punnett squares. So first of all, we have to figure out, okay, what exactly is a Punnett square? Well, Punnett square can be used to predict and to compare genetic crosses. So one of the first genetic terms we have to talk about is something called a genotype. A genotype is the genetic makeup. It's like the stuff inside you, all right? The A's, C's, T's, and G's that make up who you are. The first term we're going to talk about with this is called something called a homozygous. Now, homozygous means similar alleles. Remember, a genotype is something we can't actually see individually, so we have to use letters to represent that genotype. So homozygous, dominant, is big T, big T. And remember from before, a capital letter represents a dominant allele. Homozygous recessive is little t, little t. Remember, lowercase represents a recessive allele. We're talking about the same gene, so there's going to be two letters of the same type. You're not going to have big T and big G because there'll be two different characteristics. So we're going to deal with two letters that are the same. Now, heterozygous means different alleles, big T, little t. Well, how are you going to remember the two? Well, homozygous means similar. Heterozygous means different. Like, for example, a homosexual. A homosexual is someone who likes someone of the same sex. That homo means same. A heterosexual is someone that likes someone of a different sex, like boy likes girl. So remember, homo is similar, hetero is different. So homozygous means similar alleles, and heterozygous means different alleles. Well, so that's the inside stuff. Now we're going to talk about phenotype. What is phenotype? Well, phenotype is the physical appearance of something. You know, brown hair, tall, short, blue eyes, etc. So to remember the difference between genotype and phenotype, genotype, if you take a look at the word genotype here, genotype is, you can almost see the word gene in it, right? You can't see genes, can you? No, you can't. So genotype deals with genes, the letters A, T, C, and G. Phenotype is the physical appearance, pH, physical. All right, now let's talk about a Punnett square. Well, if we take a look at a Punnett square is basically made up almost like a window. If we go back and remember from before, each parent gives only one allele. All right, so you're made up of from mom and from dad. So each parent gives one gene. So on the outside here, you have the parent's gametes. Remember, each parent only gives one. That's why there's only one letter here. And remember, how many alleles to a gene? There's two. So one parent gives one, another parent gives the other. Now, the way it works is pretty easy. We take here, the T, simply comes down and fills in down here. And these T's right here, fill in over here. So if we take a look at the end of the day, we have big T, little t, big T, little t, big T, little t. So they're all big T, little t's. So now we got to talk about the phenotypic and genotypic ratio. What exactly is a genotypic ratio? Well, we take a look at the phenotypic ratio first, and they're all tall. Remember, big T is dominant, so big T is going to cover up the little t. And on the other hand, the genotypic ratio here, well, there's big T, little t, big T, little t, big T, little t. They're all, so you have a 1 to 0. There we go. Now you'll notice for the genotype and phenotype, you'll notice that there's not just numbers, there's numbers and a description. 1 tall to 0 short. 1 big T, little t to 0. Remember, numbers always have to have some sort of label to help give it some meaning. Well, let's do one more. If we take a look, one of the things I do want to talk about, though, is that these right here, these offspring, this represents potential offspring. So these are the possible kids that could take place. Doesn't mean that they're going to take place. Now, in this case, big T, little t are the only possibilities. But if we do a different type, let's say we do the a cross of the F1 generation, that would show something a little bit different. So let's take a look at that Mendel's F1 cross. So with this cross, here we go. To create the F2 generation, we take the parents, and we separate the alleles. So each box of the windows here represents the parents' gametes. We bring them down and cross. So we get big T, big T. We have big T, little t, big T, little t, and little t, little t. So if I were to do a genotypic ratio, I'm going to have how many big T, big T's? I'm going to have 1, right. And how many big T, little T's am I going to have? I have 2, and 1 little t, little t. Now this is, the parents here are heterozygous. And anytime you have 2 heterozygous parents, you're always going to get a 1 to 2 to 1 ratio for the genotype. Now the phenotype, well, let's take a look. Big T is tall or short. If that's going to be tall. Big T, little t, you got 1 big and 1 short, so it's going to be tall again, yep. So we have a phenotypic ratio, so the physical appearance is going to be 3 tall, or 75% tall, to 1 short. And that right there is Punnett squares.